Hi, my name is Ken Paradiso. I work for Ling-Gang at the National Institute of Neurological Disorders and Stroke intramural program, which is located at the NIH in the Bethesda campus. I'm going to be showing you the techniques for the Calyx of held presynaptic recording patch clamping. So to prepare the brain tissue, we remove the brain from the rat. We put the brain inside of this solution, which would be ice-cold, low-calcium solution, and it would be bubbling with carbogen to make sure that there's an oxygen supply to the brain at all times, and it also has to be ice-cold to minimize the amount of damage. So the brain would now be in this solution. We would take it out, cut it appropriately so that it can be put onto the block, mounted on the block with cyanoacrylate or crazy glue. Fill the chamber then with ice-cold, low-calcium solution, bubble it, place it onto the vibratome, and then cut slices at 180 to 190 microns in thickness at a frequency, blade frequency of 70 hertz and a forward advance speed of 0.01 to 0.02 millimeters per second. So that would be 10 to 20 microns per second. Once each slice is made, we transfer the slices from the ice-cold saline over to this chamber, which contains normal calcium solution. This is the same solution that we record in, and it's at 37 degrees physiological temperature. We then put the slices in here for recovery. It stays in here for half an hour to an hour before the physiology experiments are run.